Today's lecture is about protein denaturation and folding. This is from Leninger section, chapter 4, section 4. Okay, so where do we begin? The first thing that I want to say in a qualified manner is that tertiary structure is determined by amino acid sequence. This means that the amino acids contain the information needed for a protein to fold. It is worth keeping in mind that protein folding is an enormously complex process. And in spite of that, some denatured proteins, meaning unfolded proteins, can spontaneously refold into their active conformation based only on the chemical properties of their constituent amino acids, meaning those individual amino acids somehow contain the information that will tell the protein, the denatured protein, how to fold in a reasonable time frame. If we're going to talk about protein folding and denaturation, we want to think about some of the pathways involved in a process called proteostasis. That term might remind you of homeostasis, and in fact, it is quite similar. We can define proteostasis as the maintenance of our whole complex, enormous set of cellular proteins that we need to function as living organisms and keeping them at the level and function that we need. This is a complicated endeavor because proteostasis involves numerous pathways. And we not only have the folding of proteins, we have their unfolding, and we also have their degradation. So if you take some time to study this figure over here, you might notice that there's a lot of back and forth. And it's worth spending a little time looking at this and tracing some of these pathways. Notice that we start from here with the nascent polypeptide, meaning it's just being translated off the ribosome. And we may encounter some kind of folding intermediate. In fact, this is experimentally observed that we have folding intermediates before we get to the final native fully folded protein, the native protein which you see over here. Things can happen to that native protein. It can go back towards the folding intermediate. As mentioned, there's a lot of back and forth equilibrium type processes going on here. Our native protein may unfold and become a misfolded protein, in which case that needs to be addressed, or it will go and aggregate, which is something we absolutely typically do not want because that leads to a lot of neurological conditions. So the misfolded protein, however, might be handled by a chaperone and sent back to a folding intermediate stage, which in turn could be then refolded to the native protein, or that folded intermediate could go down this way. I mean, you can see that there's all kinds of possibilities here, but if proteostasis is functioning properly, we're going to avoid the kind of aggregates that you see down here, and we're going to maintain appropriate levels of our functional native protein, degrading our misfolded or old and tired proteins, and then potentially recycling them so we can make more protein. One statement that we can make is that if we lose protein structure, we will lose protein function. As noted earlier in this course, structure of our protein largely dictates its function. So one thing that we can look at is the process of denaturation. And when we say denaturation, we mean the loss of three-dimensional structure. And when we lose that three-dimensional structure, well, our protein can no longer function. So if we look at a couple of the examples up here, we can see that, for example, with an increase in temperature, we might have, ultimately, we will have a loss of function as a result of the unfolding of our protein. Another possibility is pH extreme, things like miscible organic solvents, certain kinds of solutes, detergents. There's quite a laundry list of things that can actually denature our protein. And the way we look at this in these graphs is, whether it's ribonuclease A or apomyoglobin, we check and see on this graph what the percent is unfolded. And of course, that can be anywhere between 0 and 100%. This TM here, since this graph on the left is dealing with temperature, is the temperature at which half our protein becomes unfolded. Over here on the right, we have something called guanidinium HCl. This is a denaturant, and if it is, if a protein like ribonuclease A, which we're going to be talking about in more detail momentarily, is exposed to this increasing amounts of this denaturant, it will, of course, eventually completely unfold. Amino acid sequence determines tertiary structure. We said that just a moment ago, and now we're going to look at an experiment that shows us how this was demonstrated to be the case. In order to get there, we want to understand that something that is denatured, i.e. unfolded, can potentially be renatured. In other words, it can refold, a process by which certain denatured globular proteins regain their native structure and biological activity. So if we look up here, we see the native state, we see the reduction of disulfide bonds because this is a disulfide bonded protein, and then eventually we see the complete unfolding of this protein. Now, if we renature it, in other words, refold it, it can potentially regain its native fold. We tend to say fold or conformation, and its biological activity. We want to grasp how this was established through experimentation. In other words, amino acid sequence determines tertiary structure. So there is a very famous experiment called the Anfinsen experiment, which showed what we're saying right here, that amino acid sequence has the potential to dictate the refolding process. So in this case, Anfinsen worked with the protein ribonuclease A, and he concluded that its amino acid sequence contained all the information needed to refold this protein from its linearized chain form into its biologically active native conformation. So what were the details of this experiment? Well, let's first address what is ribonuclease A. It's a relatively small protein. It's 124 amino acids, and it has 4 disulfide bonds. Its role is to cleave RNA. The experimental outline conducted by Anfinsen, and incidentally, he got a Nobel Prize for this work, was to take ribonuclease A and denature it. Not just denature it, but actually initially to break the disulfide bonds. Again, this protein has 4 disulfide bonds, and after using a reducing agent to break these disulfide bonds, to denature it, in other words, fully unfold it with a denaturant such as urea. So, 2-phase process, right? Break the disulfide bonds and completely unfold the protein. Then in steps 3 and 4, we're putting things, or he was putting things in reverse. Dialyze away the urea, allow the protein to start to refold, and then expose the protein to some kind of oxidizing conditions. In this case, air is sufficient as an oxidizing environment to allow these disulfide bonds to refold. Now let's think about this for a moment. This isn't a particularly complex protein in the sense that it is not large. It is 124 amino acids, again, not large. But it does have these 4 disulfide bonds, and there is over 100 possibilities in which these disulfide bonds could potentially reform. So that's actually a bit of a challenge. And what is it, we should ask ourselves, that ensures upon renaturing conditions, namely removing the urea and establishing oxidizing conditions, that allows these disulfide bonds to reform correctly? Because that is not a given, and that's why this was such an important experiment. When Anfinsen conducted this experiment, and he did a lot of variation in terms of order of business, the way he handled the breaking of the disulfide bonds and the renaturing, dialyzing away the urea. It was a lot of work actually that went into this, even though it seems quite simple when we put these 4 steps here. He discovered that with this experiment, ribonuclease A in a test tube would spontaneously refold to its native conformation and regain 90% of its biological function. Well, we already gave the spoiler alert. The spoiler alert was the amino acid sequence has all the information necessary to allow a protein to achieve its 3-dimensional native structure. So this experiment showed that, which is why it is considered to be quite seminal. Now, it's a little bit risky to say that tertiary structure is determined by amino acid sequence, because it worked in this case. Some proteins, it's not enough. And we'll get to that a little bit later on here. What do I mean by not enough? In other words, not all proteins are capable of folding without some assistance, folding correctly without some assistance. So hold that thought and we'll get back to it. This sets us up for a further discussion of how proteins fold, because Anfinsen showed us with this experiment that proteins can spontaneously fold, but there's still a question of how can they do it? And there is something called Leventhals Paradox. Cyrus Leventhal in 1969 stated that it is mathematically impossible for protein folding to occur if the amino acids of that protein are randomly trying every conformation until the lowest energy one is found, meaning proteins, the native fold, the native structure is thermodynamically the most stable. Let's delve into that just a bit more. So estimates vary, but amino acids of an unfolded polypeptide chain have a truly astronomical number of possible conformations to sample in the folding process. In one of Leventhal's papers, it was that the degrees of freedom in terms of amino acids sampling different conformations in the folding process could be as high as 10 to the 300. This is a more astronomical number than our imaginations can really wrap our minds around. But we can say that in this situation, if it were this big of a number, 10 to the 300 different possible conformations, it would take longer than the age of the universe for a protein to fold. So clearly that is not possible because we know proteins are folding much more rapidly than that. And the interesting thing here is we're even with this 10 to the 300 number, we're even talking on a picosecond scale, but it's still this enormous, inordinate amount of time. And while proteins vary pretty dramatically, potentially in the time they take to fold, we certainly know it's not going to be several human lifespans or longer than the age of the universe. So what could be a possible explanation for this in light of the Anfinsen experiment and thinking about how this might possibly work? Well, Leventhal's answer to this paradox was the following statement. Protein folding is sped up and guided by the rapid formation of local interactions, which then determine the further folding of the peptide. This suggests local amino acids, which form stable interactions, serve as the nucleation points in the folding process. So now we can talk about the folding process. We're going to look at this in terms of an energy landscape and how these the playing field will narrow as these local interactions predominate, and we will work our way from a very high energy state down to a lower energy, thermodynamically favorable folded protein. This requires us to address the thermodynamics of protein folding. Notice we have two different free energy funnels here. And we're going to focus our attention just on this A image for a moment. And think of it as high energy up top. And we have a y-axis here, N standing for the native conformation of our protein at a lower energy, thermodynamically stable condition. The unfolded protein will mean high degrees of conformational energy and conformational entropy and high free energy. So up here, when the protein is initially completely unfolded, there will be a lot of space for the protein to be trying different things, sampling different conformations. But we know, as we just saw, that this can't go on forever, or it will go on forever unless something happens to direct the folding process. So the proposition is that the search for the correct folded state narrows as local interactions form. Okay, so we could have the formation of secondary structure, right? We could have hydrophobic collapse. And these things together will narrow the space for the protein, and it's successively going to be having a narrower and narrower space in which to try different things out and eventually fold correctly. Now, notice there are two different scenarios here. So one scenario is that we have this smooth model. The more the one considered to be more representative, it's actually this rugged model here, where there are local energy minima where a protein may achieve some kind of intermediate state, and then somehow it's gonna have to get out of that energy well, maybe through some kind of transition state to get out of this local energy minimum. It may require the help of assistance of a chaperone, but as mentioned previously, intermediates have been observed in the protein folding process, and this is generally thought to be, again, more representative of how the whole process actually goes. Okay, so as mentioned, ribonuclease A does not require a chaperone, but many proteins actually require assisted folding. And it's outside of the scope of this course to give this any kind of detailed treatment, but I at least want to mention the presence of chaperones and indicate that chaperones are there to facilitate correct folding pathways in a whole host of ways. For example, some can bind to hydrophobic patches and assist the folding pathway from a kinetic perspective. Some can provide some kind of microenvironment that is important for allowing the protein to fold correctly. Just as a mention, chaperonins are a class of chaperone proteins. They're in the heat shock family, and they're required for the folding of large proteins. Now, some folding pathways require isomerization reactions. So this is another kind of chaperone type protein. This is one example is the protein disulfide isomerase, or PDI. We saw the ribonuclease A, and in certain instances, proteins with disulfide bonds will need the help of an isomerase to reshuffle the disulfide bonds until they reach the correct state. Another example is peptide prolyl cis trans isomerase, or PPI, which catalyzes the interconversion of cis and trans isomers of peptide bonds that are formed when proline residues are involved. The role of correct protein folding in maintaining an individual's health really can't be underestimated, because defects in protein folding are the molecular basis for many human genetic disorders. This includes, this leads to actually, defects in protein folding lead to the formation of aggregates. So one example is an amyloid fiber. Here, a protein is secreted in a misfolded state, and the key term here is inter, because generally we don't want proteins aggregating together, forming inter-protein connections. And in this case, the misfolded state will lead to the conversion of a bunch of proteins into an insoluble extracellular fiber. Another example is amyloidose diseases. These include things like type 2 diabetes, Alzheimer's disease, Huntington's disease, and Parkinson's disease, all very deleterious to one's health, and many you may note are of a neurological origin. The formation of disease-causing amyloid fibrils, as mentioned, is due to the aggregation of proteins together. So here, these proteins have a significant amount of beta sheet structure, and when the protein goes into a misfolded state, the beta sheets connect up to one another, and this is the basis of these amyloid fibrils, which is, as mentioned, a form of aggregation. Many neurodegenerative conditions have their basis in this kind of aggregation. Alzheimer's, we have the amyloid B peptide, which deposits in neurons. Parkinson's disease arises from misfolding as well. You get the spherical filamentous formation of something called Lewy bodies. And Huntington's disease is another misfolding situation that arises from a long polyglutamine repeat. Cystic fibrosis is yet another disease which has its origin in misfolding. In some cases, it's the deletion of a single phenylalanine residue, which causes improper protein folding. And then this very important transmembrane conductance regulator, or CFTR, is the function of that is impaired, and this leads to the condition cystic fibrosis. Finally, we have a whole category of misfolding called the prion diseases. This is a misfolded brain protein. And if you look here at pyramidal cells in the human cerebral cortex in their normal state versus an individual image from a patient with something called Creutzfeldt-Jakob disease, which is a prion disease, misfolding again leads to a fatal disease. So protein folding, in summary, is enormously important. Scientists are extremely interested in knowing how proteins fold and predicting how proteins fold. This can be done in a whole variety of ways. There's a lot of computational work around this. It's a huge challenge and a whole discipline unto itself. Okay, so this is our makeup lecture. Hope that was beneficial for you. And that's it for chapter 4.